researchers from various regions of Pakistan. I am delighted to share that this conference has received an overwhelming response with around 300 abstracts accepted for oral and poster presentations with 6 parallel sessions. I would like to especially acknowledge the esteemed presence of international delegates from China, Azerbaijan, and Malaysia. I am also appreciate the presence of esteemed distinguished national scientists, Professor Dr. Tariq Mukhtar, Professor Dr. Rahmatullah Qureshi, Dr. Rifat Sultana, Dr. Muhammad Mushtaq, Dr. Shah Alam Khan, and Dr. Atif Shesa Plant Protection Sciences in Pakistan. I am deeply grateful to the Sindh Higher Education for providing the major share of our financial support for this conference. I also extend heart once again for your participation and support. I wish you a productive and enriching conference experience.
Professor Dr. Abro. Now we cordially invite Dr. Mohammad Ibrahim Haskali, our esteemed conference chair and secretary, to present the Keys 2025.
Executive chair of the second event, Las Vegas University. They come from far away, especially Professor Dr. Ju from Chinese Academy of Agriculture Sciences, Professor Dr. Anis Mukhtar, she traveled along and came from Malaysia, and also the Dr. Ayura, she came from Azerbaijan. Renowned personalities of Pakistan from the various organizations, respected deans, professors, directors, officials of various organizations, virtual representatives from different countries like China, Azerbaijan, Italy, Malaysia, Egypt, Sudan, Thailand, also the representative from FAO, United Nations, representative from the media as well, my whole organizing team, and the distinguished guests, ladies and gentlemen, very good morning and assalamu alaikum. It is indeed a great honor for me to highlight the remarkable event, the second international conference on plant protection sciences. The first we conducted in 2022, that was the first event here is in Tandujam. So this happened only because of the collaboration of Sindh Higher Education Commission of the Pakistan. We are very grateful for that. So the main goal of this conference, the second ICPPS 2025, is to foster the long-lasting collaborations and the interactions among the scientists through exchange of innovative knowledge, capacity building, and diverse field from the plant protection sciences. ICPPS 2025 attracted so many contributions throughout the Pakistan as well as from various countries of the world. So we have received more than 300 abstracts in this conference. And after then, our, you can say, review committee rigorously reviewed the abstract, and finally they decided 228 abstracts. Those will be, you can say, present in 6 parallel sessions of this event. All accepted contributions has been, you can say, organized in different parallel sessions. So we have, you can say, the 6 sessions based on our 4 themes: infectious and non-infectious disease, vertebrate, invertebrate, pest management, climatic change in plant protection, weed, phanerogamic plants, and their management. Beside that, a vibrant scientific exhibitions has also been arranged for all the participants, showing the latest innovations and products in plant protection sciences. The poster gallery of the mega event, ICPPS 2025, is another intellectual attractions, giving attendees the chance to explore research and share the practice experience. To add the flavor to the scientific exchange, second ICPPS 2025 has arranged a delicious meal, and including the gala dinner, followed by the musical night and cultural show. This will be, you can say, this evening. Yeah. We hope that our esteemed guests will enjoy the pleasant and welcoming environment of Tandujam, a proud, you can say, place of Sindh Agriculture University, Tandujam. I express her team, respectable Dean, Professor Dr. Manzoor Ali Abro, as well as I am especially grateful to my worthy Vice Chancellor and Executive Chair, Professor Dr. Altaf Ali Syar, for their support and their encouragement in whole events. Yeah. We acknowledge with the sincere thanks, especially the financial assistance and the logistic support of Sindh Higher Education Commission of the Pakistan. Without that, it won't be possible. And we also, you can say, thankful for different, you can say, sponsors. Those have supported this, you can say, mega event, second ICPPS 2025. In the last, I would like to, you can say some words. Great mind don't just gather the share ideas. They ignite the revolutions in the thought. As we part of today, second ICPPS 2025, may see the knowledge shown here grow into innovations that transform the future of the agriculture and protect our the planets. Let this event not just be the memory, but the beginning of something greater. So thank you all of you, and we wish you an enjoyable and fruitful experience at our second International Conference on Plant Protection Sciences. Thank you very much once again.
Thank you, Dr. Khaskeli, for giving us a thoughtful overview of the conference. We are privileged to have Professor Dr. Rafiq Ahmed Memon, Honorable Guest and Vice Chancellor of University of Mirpur Khas. Sir, maybe we can take good remarks from you about our conference, please. Thank you. Give a huge round of applause, please.
अवल अल्लाह अलीम आला आलम जो धनी कादिर पंहिंजी कुदरत से कायम आहे कदीम वाली वाहिद वहद हू राज़िक़ रब रहीम सो सारा सचो धनी चई हमद हकीम करे पाण करीम जोड़ों जोड़ जहान जो. The Vice Chancellor, Sindh Agriculture University, Tandojam, Professor Dr. Altaf Hussain Sial, the Vice Chancellor, Uthal University, Dr. Abdul Malik Tareen, the international delegates from China, Malaysia, and Azerbaijan, and if there is any more and I forget, so please forgive me. My dear colleagues sitting out there, students, ladies and gentlemen, Assalamu alaikum. I am absolutely delighted to be here one more time because Sindh Agriculture University is my university where I had served 7 years initially at the start of my career. After serving at this alma mater, then I had moved to Sindh University, and then now I am again closer to Sindh Agriculture University, Tandojam. I would like to congratulate the organizers of this conference for putting up such a wonderful event and inviting delegates from so many countries. This is another feather to the cap to Sindh Agriculture University, Tandojam, who this alma mater is known for organizing wonderful events. I have seen Dr. Ismail Kumba always putting up these kinds of events and inviting all the people to get together and let's think about the solutions that we are facing about so many aspects of agriculture. And today's aspect is plant protection. So I'm sure that the people who have submitted their papers and the delegates who have come all the way from different countries, they will deliberate and they will provide us solutions to the problems. Conferences are aimed at, huddling together and providing solutions to so many issues that we face. In this struggle, Sindh Higher Education Commission has always been kind to provide fundings and support. So again we have support of the Sindh Higher Education Commission. So no, I'm not expert of agriculture, I'm not expert of plant protection, Basically, I am person of English literature and teaching English. So I can't give you more about these ideas of agriculture and plant protection, but I'm sure that the leadership that you have, the team that you have, they will give you more innovative solutions that can help you out. So I wish everyone a great day ahead, and I'm sure that you will be learning a lot from different papers, from different scholars, and from different researchers who have huddled together today and to provide you solutions. I am once again thankful to Dr. Altaf Sial, to Dr. Ismail Kumbar, who is sitting right in front of me, for inviting me to be part of this event. Thank you very much.
Thank you, Professor Dr. Rafiq Ahmed Memon, for your inspiring words and honoring us with your presence in our conference. We are also honored to have a Professor Dr. Abdul Malik Tareen, Guest of Honor and Vice Chancellor, Lasbela University of Agriculture, Water and Marine Sciences, Uthal, Balochistan. Sir, we may kindly request you to share your valuable remarks regarding this conference, please.
Thank you very much. My fellow Vice Chancellor, Professor Dr. Altaf Ali Sial and Dr. Rafiq from Mirpur Khas University, and my friend and my colleague, Professor Dr. Manzoor Ali Habro, Dean Faculty, and Dr. Mohammad Ibrahim Hasakhail, and the conference chair, distinguished guests, and all the international scientists who are here, and my beloved students, Assalamu alaikum, and a very good morning. It's a really honor, not only for me, but it's also honor for the Balochistan to have a guest of honor in this second international conference of the plant production, which is organized by the Sindh Agriculture University, Toronto Johnson. I really thankful to the organizing committee and especially to the Vice Chancellor of the relevant universities. As we know, recognize the importance of the agriculture and the need to safeguard it for the sustainability of the most economical and well-being of our people. This conference, like we know, is not only a merely the academic activities, but it's also a good opportunity for the students, especially who have an interaction with the world-renowned scientists who come across from the very far. I really appreciate the organizing committee that I come to know that during this conference more than 300 abstracts will be presented. So it's really a remarkable achievement for the organizing committee that 300 people will deliver or present their research, and it's a very good opportunity for the students who will be the future scientists. I commend the leadership of the Sindh Agriculture University, especially the Vice Chancellor, along with the organizers, for their tireless efforts in academic excellence, but making this larger scale international event in Pakistan of the agri. Thank you once again, and I wish the conference will be as successful, and it will be a very fruitful not only for the Sindh but for all the Pakistan. And I wish that sooner or later our university will organize the same conference, and most of the scientists, and I need help from Dr. Manzoor who will guide us, and I will make a request to the Vice Chancellor that especially pay my regards and thanks to the Higher Education Commission of Sindh, who is always very supporting to their to their local universities, whether they are in the shape of the CM or in the shape of the higher education, sir. Thank you very much, sir.
Thank you, Professor Dr. Abdul Malik Tareen, for your inspiring words and honoring us with your presence in this conference. Finally, we are truly privileged to welcome now our chief guest, Engineer Professor Dr. Altaf Ali Sial, the worthy Vice Chancellor of Sindh Agriculture University, Toronto Jam, to deliver his inaugural address of the second International Conference on Plant Protection Sciences 2025.
Bismillahirrahmanirrahim. My dear Vice Chancellor, Dr. Rafiq Ahmad Memon from University of Mirpur Khas, and Dr. Abdul Malik from University of Uthal Balochistan, from China, Dr. from Azerbaijan, Dr. Seng John Lin from China. I apologize if I have pronounced it in wrong way, because they are online foreign delegates, dear students, researchers, Assalamu alaikum and good morning. It is with immense pleasure and honor for me to welcome you all at the second International Conference on Plant Protection Sciences here at Sindh Agriculture University, Tanojam. Host this esteemed gathering of scientists, educators, industry experts, and policy makers from around the globe. From China, Malaysia, Azerbaijan, Egypt, and to representatives from FAO as well as distinguished speakers from across the Pakistan for gracing us with your presence in this conference. This conference comes at critical juncture. Our agriculture systems, especially here in Sindh, are increasing vulnerable to the twin crises of climate change and watouts, and shrinking freshwater resources are no more longer, but they are daily realities impacting our farms, impacting our agriculture, impacting our insect pests. According to conservative estimates, around 35% of the global crop yield is lost annually due to the pests, pathogens, and weeds. A figure that is often higher in developing countries, and that figure is even more in the developing countries like Pakistan and other neighboring countries. Each year, while this may reflect the seriousness of the threat, it is also underscores the need for more sustainable, climate-smart plant protection strategies and innovations. I am pleased. Let this conference be more than platform for discussion. Let it be called to action. Solutions for our farmers who are on the front lines of climate change impacts. Food security. Let this conference serve as a bridge between science and society. To our faculty members, students, and research collaborators, I encourage you to think beyond conventional boundaries. The knowledge shared here must move from lab-based appreciation in our shared journey towards sustainable agriculture, resilient ecosystem, and protected plant life in the face of climate change.
Thank you very much, sir, for your insightful words and invaluable contribution to this conference. Surely it will encourage us in the future endeavors.
We invite our next.
So thank you, and over to Dr.
Dr. Irfan Ahmad Gillar.
Me, Suman Tariq Qazi.
Warmly welcome all of you to this keynote session. Each one of them is the leader in their respective fields. Us from various countries, especially from China, Italy,
Pathology and disease management with groundbreaking contribution to agricultural sciences.
Professor Dr. Joe is currently working is the Director, Plant Protection Department, Institute of Pomology, Chinese Academy of Agricultural Sciences. He also of the Sino-Italian Joint Laboratory of Pomology and the Chief Scientist of the His research focused on the control of fungal diseases of the fruit tree and managing. Joe has also and giving a warm welcome to Professor Dr. Joe as he takes the stage for his keynote address for mayors. Professor Joe.
Okay, good morning. It's okay? Okay. And I'm very happy that to be invited by the Professor Ibrahim. And this is second time for me to be here and just to exchange some experience about our research works on the control of apple diseases and pests. Some works that are in recent 3 years. Okay, I will follow these lines and to do a simple introduction. 58 on the base of the Northeast Horticulture Experiment Station, and this is the first national professional institutions works on the pomologies. Okay, my institute is contributions to the. It's possible. May I? Look, I cannot see it clearly. It's okay. Yeah. Sorry, and since I couldn't see it clearly, so I'll have a speech here, okay? The main mission of my institute is contributions to the layout planning of the fruit planting, and also to solve the fundamental and key site technological problems of the fruit industry and all PhD students. And my department is plant protection. We have 3 groups for the pathologist, entomologist, and also for the virologist. And we know that the apples is quite important food crops in the world. It is ranks on the 3rd position of the fruit crops. It's nearly 40 billion dollars every year. And especially as also for China, is the 2nd important fruit crops. It is more or less more than 50% of the products of the world. So you can see that the apple is quite important for China. And the main regions for the apples, since it is deciduous fruit crops, it is located in the north of China. It mainly is in Bohai, around the Bohai Sea, as they showed in the red color, and also in the Louis Plateau regions, as we showed in the green colors. And what's the main problem that extreme the apple industries? First is there are plenty of diseases and insects that can contact the apple industries. Okay. And especially you can see that in the red color, some diseases and insects is still very important problems, very serious problem for China industry, apple industry. And for the blue colors, we can see that in fact the fruits, we can temporarily to control it by the paper bags. And for the black colors is not a big problem. You can see that the trunk diseases, and the canker, ring rot, and also like the Alternaria leaf spot, Malasonia brown spot, and also the Anthracnose leaf spot, and also 3 kinds of apple mice, and also the yellow aphids. This is the most popular diseases, and also is explosive in China. Okay. And the second problem is even some years ago, that the China government we have the project to try to decrease the use of the pesticides, but is still now is unable to fundamentally to solve the problem of the precise overuse. So the innovation of the theory and the technology is still need. And the third problem that is since that the people in the village areas is less and less, and so there's a very shortage of the labor force. So we must use the high efficient way for the plant protection. And how we do, first I think that the innovation for the theories, okay. In the past days we mainly based on the pesticides, and now we should to establish the new series is green APM based on the ecology balances. It's mainly based on 4 sections. One is the resistant resources. Second is for the altered ecology. And the third is the artificial strengthening of the ecology. It means that we use different green techniques to prevent and control the disease. And finally is when we must use the pesticides, we must use it in a high efficient way. Okay. And what did we do? And so I explain you something about the first is the identification and utilization of the resistant resources. So in recent years, we collect and preserve the fungi strains in more than 600, and also we established the resistant identification system. And based on this, we identified more than 100 resistant apple germplasm to the fungi disease. And also we found 15 resistant apple germplasm to the mice. And so based on this, we can provide the germplasm resources for disease and pest resistant breeding, and also for the study of the mechanism. And based on this, and also we found some very nice resistant resources, and based on this, we have the new breeding. And for my group, and we also applied 5 new varieties. They are highly and multi-resistant to 3 very explosive diseases. And also we studied some the basic research. One thing is to study the pathogenicity and to identify some, we established the mutant library of the fungi, and then we found some genes of the fungi that the pathogenicity determination. And also we studied the signal transduction process, and also studied the interactions with the plant proteins, and explain the model why they are pathogenic. Okay. And besides this, and also we study the resistant mechanism, and we find 2. Okay. This is another is the effector VNU1 is also one important pathogenicity genes. And also we analyze the microRNA that regulate the olfactory proteins of the peach fruit moth in the for locating the host. It means that these insects, how can they find the fruits? Okay, what kind of varieties they like? Okay. And also we identified and we found 2 resistant genes, GELP1 and also MDFL1. They are high resistant in the resistant varieties. So based on this, we can use it for the resistant strain breeding. And also with the new techniques for the microRNA studies, and we found some other 5 resistant genes, and these papers are under publication. And the second part is to benefit the orchard ecology. And there are 2 main things. One is we change the cultivation system from the old density orchards to the new orchards. It means that the tree is density, but the road is wide. So in this condition, we can to decrease the humidity in the orchard and to delay the occurrence of some diseases and insects. Okay. So this is quite important. And also in the recent years, we observe the difference of the natural enemies in the tillaged areas and also for the grassing orchards. So you can see there's a big difference with the grassing and new system. We can have the better ecology to control the diseases and the insects. And the third part is we have some progress on the new green prevention and control technology and products. First one is the optimization of maize technology to peach fruit moth. and we studied their genes to the 14 different fruit ornaments. Okay, and finally we selected 4 attractants. In the past, we attracted the male insects only based on sex hormones, but now we can use also the plant volatiles, so that the attraction result is much higher than before. And also we have the new spring device, so it is much better than the maize rope compared with before. And also we do a lot of research on the selection of the high efficient biocontrol of strains of the bacteria. And important is we find one bacteria that can against the bacterial diseases. We know that the bacterial disease is quite difficult to control. We cannot find more kinds of the chemical bacteria chemicals to control the disease. But with this new antibiotic bacteria, we can control some bacterial diseases in a high efficient way. And also some other new strains of the bacteria to control the fungi diseases. So for some of them, it is already being used, transferred to the company to produce it. And also we have some study, one special study is to select some bacteria at the Cerotia and can control the insects. Okay, and we spray this bacteria in the soil, and they can control these insects in a high efficient way. And this is also we obtained the invention patent, and will be used in the factory for produce this product. Okay. And the last part is how can we use the pesticide in a high efficient way. We did some work. First is the selection of the high efficient pesticides to explosive disease and pests. So this is the common work that every year we do when there's the new pesticides, and we do the identification for that. And another important work is for our group is the long-term observation of the pest and disease change. This is the task of a national platform, because in my institute there's also the observation platform for the fruit pests and diseases. And 4 tree species and 20 different pests were observed already for 6 years. So we get a lot of data for that.
It's time limitation.
Okay, yeah. And also we studied some new techniques for the spraying the pesticides with the machine. Okay, so we can see that we can spray with the drones. It's compared with the common way is high efficient and high quality, and also is economical labor equipment CV. Yeah. And this is some result that achievements for my group. Yeah, in the recent years. And for the future, we think that the innovation of green prevention and control technology is accelerating, and like the biocontrol techniques and products, and also like the microRNA pesticides, and also transfer of AI technology to the APM, the intelligent recognition of the pest and disease, and also targeted pesticide spraying. This kind of techniques is under researching. And finally, I'd like to thanks to all my colleagues, and also for my Chinese and Pakistan PhD students for the. Thank you. Yeah.
Thank you, Prof. Thank you. Thank you very much.
Thank you very much, Professor Ju, for your highly informative and engaging presentation on apple diseases and their management. Your insights have undoubtedly enriched our understanding for this critical topic. Now it's my honor to introduce our next keynote speaker, Dr. Anis Mukhtar from Malaysia. Now, before inviting her to the stage, let us share a brief introduction about her. Dr. Anis is a senior lecturer at Department of Plant Protection, Faculty of Agriculture, University, Putra, Malaysia. She specializes in insect ecology, insect toxicology, and pest management. Dr. Anis is a professional technologist Certified by Malaysia Board of Technologists, she has received several prestigious awards, including the Vice Chancellor Award in recognition of her outstanding contribution to research and academia. Further, let me share, Dr. Anis, along with her, Dr. Irfan Gilal and Aga Mushtaq earned their doctoral from University Putra, Malaysia, under distinguished supervision of Professor Rita Mohammadawang. Without further delay, please join us in welcoming Dr. Anis to share her exceptional research findings with us.
Okay, Assalamualaikum and very good morning to everyone. I think it's still not too late to wish all Muslim brothers and sisters Eid Mubarak. Okay, and I thank you for giving me an opportunity to be here to share some valuable insight of my research on population abundance and distribution of Scyphophaga incertulas, yellow stem borer. I cannot see. Okay. In Peninsula Malaysia, in particularly in looking for different planting methods and growth stages influenced its infestation. So my name is Anis. Okay. I'm from Department of Plant Protection, Faculty of Agriculture, University Putra Malaysia. So to be start with, it is a known fact that rice is a staple food for and consumed over the world in Malaysia. And in Malaysia, rice is the most important crops after rubber and oil palm. So we have 12 rice granary areas, okay, rice granary areas across Peninsula Malaysia, Sarawak and Sabah, which also classified based on the plant cultivation area, more than 4,000 hectare, practice intensive rice farming, recognized by the government in the National Agriculture Policy. And these areas plays an important role in the enhancing the food self-sufficiency level of national rice, and also enhance the food security. So this is the comprehensive overview of the rice production in Malaysia and the import trends. And as we can see here, although we produce a local production, but we still depends on the imported rice, okay, due to this low level of self-sufficiency level. And now we are about 60 to 70% SSL, self-sufficiency level, and this considered low as compared to our neighboring country like Philippines, Indonesia, Vietnam, and also Thailand. So, but we target to get around 75% in 2025, that gazetted in 12th Malaysia Plan. And there are so many factors that affect the distribution and also the national rice self-sufficiency level. And the most important factor is the pests and diseases, the disease incidence. And as you all know, there's so many pests that hinder the rice production and also reduce the yield, including rice stem borers, brown plant hopper, rice leaf folder, rice bug, and apple snails. But the most important pest for rice production and become a threat to rice industry is rice stem borers, because their infestation can cause up to 80% of the yield loss and also monetary loss. So they also can affect the rice growth starting from the 40th day after sowing. So it's very serious. So this is the overview of the stem borers species in Malaysia. The first one, we have yellow stem borer, and the color, the name of the stem borer is given by the color of the larvae. Okay, so the first one is yellow stem borer. Then we have black-headed stem borer. We call it as silo-polycrisis. Pink stem border, sesame inferens, and the last one, striped stem border, Xylo supersalis. Among all, yellow stem border or YSB or Cyphophaga incertulus is the most abundant compared to other species, especially in flood-prone rice cultivating countries. So just a short brief of introduction of rice stem borer. They are from family Crambidae, order Lepidoptera. They undergo complete metamorphosis. Consists of 4 stages, life stages: the egg, larvae, pupa, and adult. And the most damaging stage is larvae. And they bore inside into the rice tillers and feed on inner cells, where this affect the water and nutrient uptake, so the plant will eventually die. Okay, and this table shows a average life cycle of different rice stem borer species, where from egg to larvae it takes about 5 to 10 days to hatch, and from larvae they need around 33 days undergo from first instar until 6th instar, and then for pupa they need around 6 to 10 days before the adult emerge. And according to Patak and Han, the adult emergence usually happen at night around 7 to 9 p.m. And this is the key difference of rice stem borers in Asia. As you can see here, the characteristic of the adult and larval color based on the different species of borer, and also the main host. They usually prefer rice as their host, main host, but for some species like Xylo aricilius, they prefer sugarcane more compared to rice. And species like Supersalis and inferens, Sesamia inferens, can be also a future threat to corn industry and also wheat, because they have alternative hosts besides rice. Okay. Okay, all right. So I don't have meteorology, so okay. Okay. And this is the distribution of yellow stem borer species, where you can see that yellow stem borer is the most distributed in Southeast Asia. Okay. And the nature of damage and symptoms, as you can see here, it can be divided into 2. They start infesting rice starting from 40 days after sowing, especially in broadcast planting system, where starting from vegetative until reproductive or maturity stage. So for the first, during vegetative stage, they can cause dead heart symptom, as you can see here. Okay, dead heart symptoms, the third picture. And when we look into it, okay, you can see the holes on the stem and also the frost inside the damaged stem. So it become a dead heart symptom. And if the infestation, the intense feeding from larvae during 80 days after sowing, it will become white head symptoms, or we call it as whitish empty panicle. Okay, this is according to the IPM program in controlling stem borer, using rice variety that resistant to stem borer is effective. However, there is no report for rice varieties that efficient in Malaysia. There's no rice variety yet, okay, in Malaysia. But this table shows the popular rice varieties that have been planted and used by farmers in Malaysia. I think the only country that already developed rice variety that resistant to stem borers is India. They've produced several varieties, and Ratma is one of the variety, if I'm not mistaken. Okay. These are the factors influencing the distribution of rice stem borers, and integrated pest management is the best way so far to control. However, the chemical is not really effective as of now, because the contact of the chemical with the larvae is become shortened, because the larvae will bore inside the stem. Okay, so we have to look into the threshold level before we apply any chemical insecticide, so to prevent any further resistant issues. So light traps are using to monitor the population of stem borer in the field. Okay, the left side, okay, that is the bucket light trap that have been improvised and developed by my team, okay, and compared to this picture, okay, the net 10 light trap is a common light trap. The bucket light trap is very efficient and effective to collect more adult at night. Okay, so this the objective aim to assess the population and to know the species occurrence of yellow stem borer, particularly in Peninsula Malaysia, and also to monitor the population abundance of major rice stem borer, different planting methods and rice growth stage. So for the first experiment, we select 10 rice granary areas with 3 locations for each granary area. The location must be more than 5 km apart of each other, and so the total sampling size is 30. And for the sampling, we use 2 bucket light to collect the adult at night, and sampling at night 7 to 10 p.m. 3 hours at 80 day after sowing paddy. So this is the top view of bucket light trap. In the paddy field using the bucket light trap. Okay, these are the locations, the 3 locations for each granary area, and as a result, there are 4 species that have been collected and identified across the 30 location, sampling from 30 locations in Peninsula Malaysia, whereas yellow stem borer contribute the highest abundance compared to other species. And when we look into details, so based on the 10 granary area, the population of yellow stem borer is highest in Aida Barat Laut Selangor, in Selangor. Okay, and so it is proven that as in Setulas, the yellow stem borer is dominant, aligned with the literature that often cites this species as primary pest in Asian rice system, and a relatively lower abundance of other species like Xylopolychrysis, Sesamia inferens, and Schizophaga inotata suggests that they may play secondary role of infestation, or maybe more localized or seasonal based on the region. And the dominance of the YSB is due to several factors, including highly adapted to tropical rice ecosystem, and according to Khan and Joshi, Silo and Sesamia inferens prefer drier environments and commonly associated with upland or less irrigated rice systems. And the last one, Schizophaga inotata, even though they have same genus with Incertulas, they often prefer specific rice stage and may not be well synchronized, or maybe they face higher natural mortality due to effective parasitism in this region. And this is the reason why Aida Barat Selangor is contribute highest number of YSB. The first one because of the continuous cropping. So the YSB get continuous supply of food, and climatic condition, Selangor is warm and humid tropical climate, allow for multiple generation of YSB and pesticide misuse. And the second experiment, I use Barat Laut Selangor, but 2 different planting system at different growing stage. So it can be proven that broadcast planting system contribute the highest number of abundance of YSB, and 100 days after sowing for broadcasting system, giving the high abundance of YSB population. And this suggested that the highest infestation stage may vary depending on environmental conditions, rice variety and farming practice. And this because for broadcasting system, the larvae of YSB usually use circle track to move from one tillers to another. So when the distance is short in broadcasting system, they will cause severe infestation compared to transplanting. And this supported by Kumba and Singh, that broadcast can cause up to 95% infestation compared to other cultivation system, including transplanting, drum seeding, and system of ASRI, system of right infestation. So as a conclusion, as in Setulas, showed significantly higher abundance than the other stem borer species. And for a broadcasting system, 100 DAS shows the highest number of infestation, and it is suggesting that population pattern influenced by growth stages, planting methods, and also regional conditions. For recommendation, breeding and development of resistance variety is very important and essential for sustainable stem borer management in Malaysian rice granary. So this is one of the publication by my team. I think that's all. Thank you. Okay.
Thank you, Dr. Anis, for that excellent and insightful presentation on the yellow stem borer that is a global pest, including Pakistan and Malaysia. So your work is truly commendable.
Our last keynote speaker for this section is Dr. Inaugural from Azerbaijan. Dr. Inaugural is a head of Proteomics Laboratory, Institute of Molecular Biology and Biotechnologies. Yes, please.
Hello everyone, Assalamu alaikum. It's a very great pleasure for me to be in here. It is to have this experience, and I am very thankful to all the organizers and especially to Dr. Ibrahim Khalid for his invitation, and for everyone, for all the organizers to make my presence here possible. Today I will present I will give information about the research work which we are doing in Azerbaijan, and so here is the brief information about our institution where I am working, and I work in the Institute of Molecular Biology and Biotechnologies. Our institute is operating under the Ministry of the Science and Education of the Republic of Azerbaijan, and the structure of our institute has several laboratories, and as it was mentioned before, I am leading the Laboratory of Proteomics currently. So, and our institute is located in the Baku city, so we can enjoy this beautiful view of Baku just nearby our institute. So next time, welcome to Azerbaijan, and we will be happy to see you in Azerbaijan. And now I will give information about our research work. So my research is mostly related to photosynthesis, and as I am a molecular biologist, and we are trying to understand this process in molecular level. So I think we all know about the photosynthesis, about the importance of photosynthesis on Earth, and yes, it is important to convert light energy to chemical energy through the photosynthesis. And photosynthetic organisms, we know that they can live everywhere and they can adapt to different conditions. For example, some of plants can live in the stable, constant 20 hours light, 20 hours dark conditions, but other plants just live in conditions with very small, very few amount of the light. But it means that it is actually good news, because it means that plants can adapt. It means that they are able to make some mechanism to protect themselves and to live in their surroundings. And what we are trying to do, we are trying to understand these processes, and we are trying to understand the effect of different abiotic stress factors like light stress, salt stress, and some other stress to photosynthetic apparatuses. And we try why it is important, because if we know that plants can adapt, by learning these mechanisms, how they can adapt, we can increase their crop productivity, and we can give them new ways, new biomarkers to make plants more resistant and to have more productivity from crops and from other plants. So now I will take you in our playground, so the organelles with which we are working. And here you can see the leaves crop sections, and inside, of course, here you can see the you can see the chloroplasts, the main organelles, which are important for the process of the photosynthesis. And if we look at the cross section of the chloroplast, so here we can see the thylakoids, and thylakoids, we know that main photosynthetic complexes are located located on the thylakoid membranes. And if we look at this like a cartoon view, so here we can see the structure of the thylakoid membrane, and thylakoid membrane consists of different components, big multi-protein components, which are very important for the photosynthesis processes. And here red circlet is the light harvesting complex proteins, which first captures the sunlight and then they send the photosynthetic centers and cytochrome B6F complex then to the PS1, and these complexes are located in the, as you can see, on the thylakoid membrane. So in our research, we are using thylakoid membranes, we are extracting thylakoid membranes, and for further investigation for learning the impact of the light stress, or as I mentioned before, salt stress, drought stress, and other additional abiotic stress factors. Today's my presentation is related to the light factor, and why we are talking about this, because we know that during the day and even during the different different seasons, the light intensity, light quality is very often changing. Even in the same tree, we can see that some leaves are very strongly affected by the light intensity, but others have just shade, or because of the wind, they get less light. And during the day also, we can see from this graph that light intensity from the morning time to the night time is just flexible, and this light intensity is changing. And different light intensity have different impact on the plants, on the thylakoid membrane, and the efficiency of the photosynthesis. And plants are just amazing organisms. They can have a different mechanism to protect themselves. They have just short-term mechanisms. For example, they just start in a few seconds. They switch on some mechanism to protect themselves, and then they again switch off this mechanism and turn on another mechanisms. And we said that there is sometimes impact of the high intensity light, and in other cases the intensity of the low intensity of the light. And it is the question, it is that how do the plants prevail over excessive light, because dangerous. But when we talk about the high light intensities, it is very dangerous, because there are a lot of photons coming, and they are using for light reactions, but so it makes a problem, the real problem for the plants. And here just I shortly illustrate the mechanism which plants develop for the protection from different intensities of light. Here in the left side, we can see the excited energy is just distributed between two photosystems, like part of the antenna is going. Light intensity is too much if it is in exit, and they cannot use this energy for photochemistry, these photons for photochemistry. So in this case, plants develop additional mechanism, which is take main part, the main short-term reaction response. It is the non-photochemical quenching. And what is the non-photochemical quenching? When the exit light is just dissipated like a heat, and it is not used for photochemical reactions. And here we can see the different mechanisms and different structures which are working on these two mechanisms. So here is different plans and different objectives that we are using in our laboratory. In addition, during these all processes of the acclimation to different light intensities, they also occur the processes of phosphorylation. And phosphorylation is like a kinase and different kinases and different phosphatases are working on these processes. And here we try to understand, in this research, we try to understand how the fastest component of non-photochemical quenching is correlates with the phosphorylation of processes, and the role of the PSPS protein, which is important for this process of the MPQ, is related to this process. And for this research, we use different tools and different objects. So we used actually actually, in most of our research, we used plants like wheat, rice, Arabidopsis. And here in this research, I show the result with the rice plants, wild type, and PSPS no-count plants. And another one is the Arabidopsis plants, also wild type, and PSPS no-count plants. In this case, we call them MPQ plants. And we are using different methods like gel electrophoresis. You can see here HPLC analysis, and we try to integrate different methods like Western blot analysis, histochemical staining, also fluorescence measurements, and of course the data analysis. And here just I will give brief informations about which results we gained with these studies. And here you can see in this slide our study related to the effect of the highlight intensity on the phosphoprotein of the thylakoid membranes. And what we did in here, after the light illumination, after highlight illumination for 10 minutes and 1 hour, we investigated the phosphorylation level of different proteins, and we could observe the increase in the phosphorylation level in the PSPS co-mutants in compare with the wild type plants. And then we established another technique which we used for first time with phosphoproteins. It was the technique which we call the direct transfer from the membrane, from the bean gel to the membrane. So here we can see in the right part, you can see this technique where we can see the bean gel, and in the right side we can see the result with Western blot analysis. Why this is important? Because even if in traditional way, we can see that just the total amount of the protein, of the core proteins, of the light harvesting complex proteins. In this way, we can see the phosphorylation level of the individual proteins, and we can see in which complexes they are phosphorylated more and how it depends on the intensity of on the treatment time. So that is why we established this method and tried this method in our works. And here we present the results related to the wild type and PSPS co-mutants with the same light intensity treatments. And as we could observe from here, the phosphorylation level in the PSPS co-mutants was higher in comparison with the wild type plants. And next we were checking the individual light harvesting complex proteins, and from light harvesting complex proteins, the phosphoproteins are LHCb1 and LHCb2, and we check their phosphorylation level in different highlight treatments. And here was interesting result after the wild type treatment for 10 minutes, because we could observe the high level of phosphorylation in this case, and but in the case of the PSPS co-mutants, it still was phosphorylated in the conditions like highlight treatment for 10 minutes and 1 hour. And we use the same technique with bean page in the same plant to check the individual proteins in different pigment protein complexes. Also, one of the important proteins which we are interested in photosynthesis, so this is the CP29 protein. Here, the direct transfer Western blot results show it that here the level of this protein was increased after highlight treatment, and we suggest that CP29 is regulating the distribution of the excitation energy between photosystems, and this is one. Its mobilization led to the decrease of this protein's level. So also we check the effect of the low light intensity of the phosphoprotein of the membranes. For this case, if we before used highlight intensity, but here we use just low light intensities, and we check how this phosphorylation level change under the low light conditions. And here also we can see the results with just traditional Western blot analysis and with the direct transfer from the bean page to the membrane, and higher, more significant differences we could observe in the case of the PSPS co-mutants. And next we check the level of the phosphorylation of the LHCb1 protein, and interesting result was with the low light treatment in the PSPS co-mutants, because the level accumulation of the phosphor-related LHCb1 proteins was very high in the case of these mutants. And so light-induced phosphorylation of the photosystem II core proteins and also light harvesting complex protein is enhanced overall in our mutant plants, and we suggest that this may cause it by the higher level of the ROS production in these plants, and also we suggest that also the PSPS protein impacts the kinase and phosphatases work, their networks, and this way they have just some difference in the phosphorylation levels. Here just the result of this, okay, so here just staining results for checking the ROS levels, so peroxide and hydrogen peroxide levels, and we could observe here, as we suggest, we observed here a higher level of the accumulation of ROS in the case of the mutant plants. Also the pigment analysis, we did some pigment analysis with our wild type and mutant plants, which also showed the differences in the composition of the pigments of the PS2 enriched membrane fragments, and mostly increasing level of the neoxanthine, viloxanthine, leucine may improve the plant's ability to dissipate excess light energy as heat through non-photochemical quenching. And also we observed the increased content of neoxanthine in the PSPS core mutants, which may cause by the conformational change in these plants. Also another way, experiments which we are using, which we did in this research. This is the measurements of the NPQ kinetics after the different treatment times, and here we show all our wild type in the left side and in the right side the mutant plants, and of course as we suggest we had a much lower level of the NPQ magnitude in the case of our mutant plants. And also, interestingly, in the wild type case, after the highlight treatment for 10 minutes, we could observe the slowdown of the NPQ relaxation, and it is known that NPQ slowdown is relaxation may be important for the crop productivity, and there is some research works related to this. And based on all these results, we suggest the NPQ induction and relaxation kinetics is highly conceivable, and that phosphorylation of LHCb1 and LHCb2 proteins are closely related to the rapid relaxation of the NPQ, as well as the higher rate of the photosynthesis. This is just a model which we proposed for the, these processes in the case of the presence and absence of the PSPS protein. And this is just summary, and we actually, our laboratory is international laboratory of the proteomics, and we collaborate with some other international laboratories, and this work we did in our institute with collaboration with the Busan National University, which is in Busan, Republic of Korea. And I want to thank my late scientific supervisor, Ismail Zulfikarov, our head of our institute, Irada Husseinova, and from Korea, from the Laboratory of Molecular Biology, Professor Chunan Lee, and all the lab members to make this research possible. And thank you for your attention.
Thank you much, Dr. Ayonara, for sharing your valuable expertise with us. Indeed, it was an impressive and thought-provoking presentation. Thank you. Now, it is my honor to invite our chief guest, the esteemed Vice Chancellor of the Sindh Agriculture University, Tandojang, Engineer Dr. Professor Altaf Ali Siag, to kindly present the conference sheet to our distinguished guest of honor. So please. So I request Professor Dr. Abdul Malik Tareen, Vice Chancellor, Lasbela University of Agriculture, Water and Marine Sciences, to collect his conference shield from our worthy Vice Chancellor. Thank you. Thank you very much, sir. Apply. With this, we concluded our keynote speeches of this session. Thank you very much.
Here we have two important for their remarkable participation. There is a tea break at 12 a.m. and second keynote speeches will be resumed at 12:20 at same auditorium hall.
For the official inauguration of the conference, exhibition and poster gallery. And Professor Dr. Bhai Khan Saab is requested to accompany the honorable guest towards the conference exhibition and poster gallery. Thank you very much. So please be seated on your seats. First, let the honorable guest to proceed for the exhibition gallery. Okay, sir. Thank you.
Bilal, we welcome you in this keynote session where we are renowned teachers, researchers from Pakistan and various countries of the world such as China, Egypt, Azerbaijan, Italy, Malaysia, Sudan, Food and Agriculture Sciences Organization of United States will represent outstanding research findings.
So our first speaker is Professor Dr. Rifat Sultana. She is a professor of zoology at University of Sindh, Jamshoro, specializing in entomology. She earned her PhD in zoology and have two postdoctoral research programs at USA and China. Her research focuses on biodiversity, insect systemics, molecular and biological control, especially on Orthoptera. So without further delay, I request Professor Dr. Rifat Sultana for her presentation. Prof.
नहीं सनी यहाँ से मुझे मुश्किल ये रखा दो बस वालेकुम सलाम छोड़ थोड़ा देर कर हटो मैं इसको खोल दूँ
कंट्रोल करना नेक्स्ट आप बता दीजिए मैं दूसरे करता जाऊंगा या इसे करो
नहीं मैं खुद कर लूँगी
इसे नेक्स्ट करते जाना आप दोनों से
हाँ मेरी प्रेजेंटेशन डाल दी आपने इसमें फिर यूएसबी दो ये कनेक्टेड है सर मेरा इसके साथ मीडिया
नहीं ये नहीं हो रहा ना इसलिए हम नेक्स्ट कर दीजिए डॉक्टर तो इसको
छोड़ दो यार इतना तो तुम लोग ट्रबल में डाल रहे हो मैं चश्मा लगा के पढ़ लेती हूँ हाँ छोड़ो बस हाँ देखती हूँ सही इट्स ओके हाँ सही है बिस्मिल्लाहिर्रहमानिर्रहीम ऑनरेबल प्रोफेसर अब्रो डॉक्टर इब्राहिम डॉक्टर इमरान खत्री all the distinguished scientists and the scientists coming from the abroad. Assalamu alaikum, welcome all of you, and very good afternoon. And all the scientists, research scholar, and my dear student, assalamu alaikum. Today I have a talk on the current classification and the recent innovation in the Insifera Arthroptera with the emphasis on the phylogeny. Next, please. नो नहीं, मुझे थोड़ा ट्रबल आ रहा है इससे देखो, क्योंकि वो मेरा आई साइड पे इशू है। नहीं है ना क्लियर? ये दे दो बेटा, मुझे दे सकते हो यही वाला? हाँ मेरा है सनी प्लीज़, क्योंकि ये क्लियर नहीं हो रहा मुझे, जल्दी प्लीज़। मुझे यहाँ रख दो, मैं कर दूँगी इधर में वो USB बंद क्यों किया आपने? USB लगा दिया आपने?
उधर मैं नेक्स्ट करता जाऊंगा।
छोड़ दें आप, मैं इसको कह दूंगी बच्ची को, नो प्रॉब्लम। नहीं ठंड हो जाओ। पहले ही कर देती हूँ, मैंने आपको पहले ही कहा था। नाइस, ठीक है, मैं कर लूँगी। Sorry for the inconvenience. So actually I will talk on the current classification and the recent invention in the Incifera arthroptera with emphasis on the phylogeny. As all of you know that there is the era of the DNA barcoding. So there is a need how we classify the species on the basis of the DNA barcoding. So the overview of my talk is the overview of the Incifera, what the importance of the Incifera in the arthroptera diversity. In the results, I will try to give you a little bit knowledge about the current taxonomic framework of the Incifera with the addition of the morphology, how is affected with the help of the molecular approach. And what are the limitations to adopt this strategy in the future. Next, please. So, no, previous. So, first of all, what are the arthroptera? Because the Incifera belong to the arthroptera, so we have to know little bit about the arthroptera. Arthroptera actually is the largest order in the class Insecta after the Coleoptera. These mostly are the terrestrial and they are very dangerous to the crops. These are the phytophagous, these are the predators, some are omnivorous, some are the carnivorous. And this, their nymph resemble with their adults. Mostly they oviposit in, oviposition is the common while the parthenogenesis is the very rare. And in here, if you see this table, so this is the valid classification of the arthroptera. At the globally levels, up to date, there is the description of the valid species that is the more than 30,000, but still there is the need of the addition of the some species on the basis of the morphological, on the basis of the DNA sequence. Almost the species they were identified on the traditional taxonomy. So actually my basic aim to talk here is that now the era is a change, so now we switch to us and we can combine our efforts with the morphology as well as with the addition of the DNA barcoding. So if you view these slides, so you can see there is the huge difference between the Incifera and the Califera. But actually the arthroptera, they divided into two basics, the suborders. The first one is the Inciferine and second one is the Califerines. In the Inciferine there is the cadet and the cricket, there is a common member, while in the Califerines you are familiar, the grasshopper and the locust, they pose the disaster to over the agriculture. So there is a lot of work has been done by me